Hello, good people of Anthro 101. This begins two lectures on genetics and evolution and inheritance, etc. I always say they're going to be short, but these ones I know are probably not going to be short. So sit back, relax, grab a beverage, water. You need to stay hydrated, and let's get into it. I always forget I can use my fancy pen, so maybe I'll do that. All right, let's start with the cell. Now, this is going to be a lot of information. You're not going to have to know absolutely everything. I will try to emphasize the things that I want you to know for exams and so forth. Okay, so there's two basic kinds of cells, prokaryotes and eukaryotes. Most, if not all, multicellular life are eukaryotes. So that's really what we're going to be focused on here. But the one on the top here, this one is a prokaryote. A karyote has to do with the nuclear envelope in the nucleus of the cell, which I'll get to in a second. And so U means true. So eukaryote, true or good. Eukaryote is a cell with a true nucleus. These prokaryotes, they don't have a nucleus, which means that the DNA, which is right here, is kind of free floating in the cytoplasm of the cell. Those are usually unicellular organisms like bacteria. But it's really the eukaryotes here that we're most interested in, and that would be this guy. So eukaryotes have a nucleus, which you can see here, and inside the nucleus is where the DNA resides in a eukaryote. So plants and animals are eukaryotes and so forth. As far as parts of the cell, I'll circle the ones that you really need to know. I mean, there's a membrane around the cell, which should be sort of familiar from, I don't know, basic high school biology. Then you've got the nucleus, so you should know what the nucleus is, and this here really is the nucleus of the cell, as you can see. And inside the nucleus, you've got your DNA, which is wound up in the form of chromatin. It's basically like spaghetti-like strands. It's like a bowl of spaghetti here in the nucleus. Okay. Now you've also got these things called organelles, which are like, think of them like miniature versions of organs in your body, in the sense that they have a specific function that helps the cell to do what it needs to do. So I want you to know about mitochondria, which are these kidney bean shaped things floating around in the cell. Those are considered the powerhouses of the cell. There's many mitochondria in each cell, and they have their own DNA, which is fascinating, and it's totally separate from your nuclear DNA. And if we were in class, I'd ask if anybody knows why that is, but the hypothesis is that these were once their own sort of unicellular organisms that were swallowed up by, the first or some of the first eukaryotic cells. And they have sort of a symbiotic relationship because the mitochondria gets nutrients that it needs from the cell, and kind of in return, it provides energy for the cell to function. You also should know I'm probably not going to ask you about endoplasmic reticulum. I'm not sure though. So no endoplasmic reticulum. That is this rough endoplasmic reticulum. All this stuff here. That is what the ribosomes are on, and the ribosomes are important. Those are these little dots here. And those little dots are sort of the factories where proteins are made. Okay. And that's going to be important because the primary function of DNA is to carry instructions for making proteins. Something that I did not mention specifically is cytoplasm. And cytoplasm is just the fluid kind of matrix inside the cell that all these organelles are floating around in. So there's just like a larger kind of version of it. You can, that'll be helpful for you when you're looking at the PDF of this. But again, you want to know the nucleus, you want to know endoplasmic reticulum, you want to know ribosomes, you want to know the mitochondria. Mitochondria is plural, mitochondrion is singular. And those are the most important kind of parts of the cell that we'll be kind of coming back to. So DNA, what is it? What is it good for? DNA is deoxyribonucleic acid. And DNA is essentially made up of, it's like a ladder. So on each side of the ladder, what kind of provides the backbone, I guess, are these this chain of molecules made out of sugars and phosphates. So that's what the S and the P are here, sugars and phosphates. And that just provides structure. But the what forms the genetic code is the sequence of these are called nucleotide bases here in the middle that form the rungs of the ladder. And so reading the genetic code, you kind of read from one end to another of what these nucleotide bases are. There are only 4 nucleotide bases. There's adenine, thymine, guanine, and cytosine, and they're abbreviated A, T, G, and C. Now, A and T always bond together, so you always get these two together. So if you have an A on one side, you have a T on the other. If you have a T on one side, you have an A on the other. And then G and C always bond together. So you get a G on one side, you get a C on the other. If you have a C on one side, you get a G on the other. Okay? How do you remember that? You can come up with whatever way you want to remember that, but I remember that at forms a word, okay? But g doesn't really form a word, but just remember that at is one of the ways that that they bond together, okay? Now, a gene is nothing more than a stretch of DNA, a certain area in DNA that codes for a particular protein or a series of proteins, okay? When people say, oh, there's a gene for that, the gene for eye color, the gene for height, the gene for sexual orientation, or what, it's become like real big to try and find, there's a gene for that. Well, there's rarely a gene for that. There's usually genes that have an effect on something, but it's relatively rare for there to be a single gene to control a single trait. Right? It's I would go so far as to say it's extremely rare, but it does happen. Now, we all, except for certain genetic kind of anomalies, where maybe there's a section of the DNA that has been snipped out or deleted, we all have the same genes. I know that sounds counterintuitive, but think of it this way. Think of DNA as like a blueprint for a house. And a gene carries instructions for a particular section of that house, right? So imagine that there's, a gene for the master bedroom and a gene for the bathroom and a gene for the entrance and a gene for, the kitchen, okay? We are all made up of the same things, right? So for the most part, 99.9 blah, blah, blah % of people are going to have two eyes, a nose, two ears, blah, blah, blah. Like, we have the same basic structure. So we have the same genes. Now, where we're different is our kitchens may look a little bit different. Our bathrooms may look a little bit different. Our master bedrooms may look a little bit different. And those differences are alleles. So an allele is a different version of a gene. Okay, so like for eye color, we all have eyes. All of our eyes have some kind of color to them in the iris. But there are obviously different colors that exist. And what makes the difference is not different genes, it's different versions of those genes. Okay, and those versions are called alleles. So, for example, what you see over here on the right are 3 alleles of the same gene, of a gene. Sorry, it's hard to write on my laptop screen, but okay. So, this yellow one here, this may be the one that's most common. Let's say this is the stretch of DNA in this gene, and this is the most common sequence there. But then there are alleles where there's differences. Like, for example, here there's a difference, right? This A, which is sort of the original, if you want to say, nucleotide there, has mutated, has changed to G. And that G is shared by both the red allele and the blue allele. But then there's another area where they're different right here, where the yellow and the red both have an A. Whoops, whoa, I didn't even know I could do that. This is crazy. And the blue has a T. All right, so that's a mutation. So what makes this blue one different is that it not only has a G instead of an A, but it also has a T here instead of an A. Okay. Okay, so within a gene, there are areas that code for proteins and there are areas that don't code for proteins. And in fact, throughout all of DNA, there are areas that don't code for proteins. The ones that do are called coding regions. Those coding regions have genes within them, but even genes may have non-coding regions within them. Non-coding regions do not either do not contain genes in them, or they're sections of genes that don't specifically code for proteins. I know it seems a little bit confusing, but just think coding means it carries some instructions for a protein. Non-coding means it doesn't, okay? However, these non-coding regions, this is just kind of interesting. I don't think I will ask you about this. The non-coding regions used to be what scientists called very technically junk DNA. And because they didn't know what it did. And in fact, the vast majority of the human genome is made up of what used to be called junk DNA. We now know that that junk DNA is actually really important, and one of the functions of the junk DNA can be to provide regulatory regions. So if you think about a gene that here's an example. If you are, an adult and you can drink milk and you have zero kind of gastrointestinal repercussions from that, you are actually, according to the research, in the minority of humankind. So the vast majority of humans are lactose intolerant as adults. And I'll go into that in other lectures. I can go on and on about this because I was going to do a PhD on this before I switched. But and that's all because there is one gene. This is one of those rare circumstances where one gene can have a real serious impact on a trait. There's one gene, LCT, that codes for the enzyme lactase, and you need that enzyme to break down the main sugar in milk, which is lactose, so that you can digest it. Now, it turns out that the difference between people who are lactose intolerant and those who are what they call lactase persistent is not a difference within the coding region, it's a difference in a regulatory region. And the regulatory regions act like either switches or like volume knobs. You know, you can turn the volume off, you can or you can turn it on, and you can turn it up, you can turn it down. And so turning something up is called up-regulating, turning something down is called down-regulating. And so there's a section near that LCT gene, or possibly within it, I don't remember, that is a regulatory region, and a certain mutation of it breaks that regulatory region. Now, usually when you become an adult, that regulatory region tells your body, stop producing this enzyme because you're not breastfeeding anymore, hopefully, as an adult. However, in some individuals, there's a mutation or a series of mutations that breaks that regulatory region, so your body keeps producing it, even as an adult. So regulatory regions can be really important. Here's a kind of a closer up schematic of what a gene looks like. So this section here is the gene. There are non-coding regions at the beginning and the end, for example. The promoter region that tells, basically it says, all right, this is where we're going to start producing this protein. And the terminator region says this is where we're going to end producing this protein, okay? And then in between, you have a sequence that codes, that carries instructions for making a protein. Now let's look at that a little bit more closely. Here's the promoter area, okay? And then somewhere over here is the terminator. I'll be back. Boy, that's terrible. That says TERM period, sorry. Within that, okay, within the coding sequence, there are still non-coding regions called introns, okay? So these exons code for amino acids. And amino acids are the building blocks of proteins. These intron regions don't code. Okay, so when a gene is being read by the sort of your cellular machinery and it's going to be turned into a protein, what happens first is these introns are spliced out. Remove introns. So you'll notice that the exons here are like yellow, except for like the start and the stop ones, which are red, but yellow, 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 right? So that's what you have here. Okay, the exons, the introns are gone, right? So it's this thing that moves on here to protein synthesis. All right, we'll come back to protein synthesis. Chromosomes. Chromosomes are made up of DNA. First, okay, in the nucleus, so here's the nucleus of the cell. In the nucleus, you have these noodles that are made up of chromatin. Chromatin is basically DNA that is wound around histone proteins. All right, just think of it as convenient packaging. All right, because there is a lot of DNA in a single cell, a lot of DNA. So rather than have that DNA just be like, whoa, I'm floating around willy-nilly, it gets sort of prepackaged into chromatins, right? So the DNA gets wound around these histones, and the histones, kind of bunch together, and then you get these chromatin fibers, all right? And that's how it spends most of its time in the nucleus. Now, during cell division, which we'll get to later, whether we're talking about mitosis or meiosis, again, I'll talk about that in a second, these strands of chromatin, then they wind themselves up even more. They package themselves in a more particular way called chromosomes. Okay? So a single strand is a chromatid. A single strand of chromatin is a chromatid. Okay? And chromatids wind up and become chromosomes eventually. Now, as far as chromosomes go, humans have 23 homologous pairs. Homologous means that a pair is made up of the same. All right, so let me just, can I do this? It's not letting me draw. Okay, so let me just draw something. So let's say here is chromosome 1, chrome 1. And let's say this is from your mom, right? Your mom gave you a particular, she gave you a version of her DNA in chromosome 1, but you also have a chromosome 1, I'll just put CH1, from dad. Now going back to the blueprint analogy, these are called homologous because let's say here's a section of chromosome 1 that codes for your master bedroom, and the same section of your dad's codes for a master bedroom. God, I'm sorry, that writing on here is really difficult. Okay, so they're homologous because they carry instructions for the same things. Now the instructions may differ, right? Your mom might have a really luxurious master bedroom with a walk-in closet and everything like that. And your dad's may be much simpler. Much like, let's be a little bit more real about it. Maybe your mom has brown eyes and your dad has blue eyes, right? Well, the location of those instructions is going to be on the same place of their chromosomes, but the instructions may differ, okay? So you have 23 pairs of these homologous chromosomes. Okay, so that means you've got chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, and on and on and on. And you've got one of those from mom and one of those from dad, okay? Within those, so if you do the math, 23 times 2, that's really hard, I know. It makes 46 total chromosomes. 23 pairs, 46 total. 23 pairs of shoes, 46 individual shoes. Whoa. Now, of those 23 homologous pairs, 22 of those pairs are what we call autosomes. An autosome is any kind of chromosome except for a sex chromosome. A sex chromosome is an X or a Y chromosome. And, when we talked about gender, we talked a little bit about that, sex and gender. So typically, although we know not always, but typically, X and Y carries instructions for a male, for the development of male genitalia and so forth, and internal reproductive organs, etc. An X and an X carries instructions for a female. Okay, so typically only males have a Y chromosome. Now so this is the 23rd pair, the sex chromosomes, either XX or XY usually. Okay, so the other 22 pairs are called autosomes. This just shows a different version. This here is called a karyotype. Let me see if I can do this. Karyotype. It's a way to look at chromosomes under a microscope after staining them, so you can see sort of individual regions. I won't ask you about karyotype, but in case I mention it, you should know. And then these two things are schematic ways of viewing a chromosome. In the middle of each chromosome, they kind of pinch together, and that area is called the centromere. And then there's a shorter arm and a longer arm of the chromosome. And the shorter arm is called P, and the longer arm is called Q. Again, I'm not going to ask you this. But just for future reference, if you're interested in this stuff, one of the ways that you can talk about the location of a gene in the genome, you can use this chromosomal location notation. So if you're talking about a particular gene here, you can say it's on chromosome 7, it's on the Q arm, so that would be this one here, And then you've got a location, 31.2, or region 3, 1.2, blah, blah, blah. All right. Yeah, so I'm just going to move on here. Cell division. Okay, there's two types of cell division that we're going to talk about. One is mitosis and one is meiosis. Hold on a second, I need to hydrate. Okay, so most of the cells in your body are called somatic cells. Aside from your reproductive cells, aside from like your sperm and your eggs. And those cells need to replicate, right? Because cells die and because people grow. You're hopefully not the same size as you were when you came out as a baby, because either that means you're really tiny or your mom hates you because that was a terrible, terrible labor. So I'm sure your mom doesn't hate you. But needless to say, you've grown a lot since you were born. And in order to grow, your cells have to divide, right? You have to make more cells. And also you're sloughing off dead skin cells all the time, that's what exfoliation is all about. If you care about your complexion and whatnot, you got to exfoliate. If you have dandruff, that's natural exfoliation, I guess. So the cells divide, and mitosis is the process by which cells divide, and they make copies of themselves. All right. So they are complete 100% copies. So what happens? You don't need to know the names of all these phases. But essentially what happens is you've got the DNA here wound up in these, chromatin strands. And then as you're going into mitosis, they'll bunch up into chromosomes. And those chromosomes, the reason why you have, so originally you would have a chromosome looking like this, okay? But what happens is the DNA replicates. So you kind of get the original and then you get a copy. And that's why the chromosome has this X shape. And it's bunched together at the centromere. Okay, so that is copied. DNA. All right. So the DNA is double-stranded. It kind of unzips and then it makes copies of itself along one end. Because recall, if you have an A, it bonds with a T. If you have, a C, it bonds with a G. If you have a G, it bonds with a C. So all you need is like one end to kind of make a copy of the DNA. You only need to know one part of the code, and then you can figure out the rest, the other half. So here, these things are called sister chromatids. All right, because they are they're really like twins. They're identical. All right. So then the chromosomes they line up at the center of the cell, and then the cell sort of pinches itself apart here, and it pulls those copies apart to different sides, and so you end up with two new cells that have the exact same DNA and the exact same amount, 46 chromosomes. All right, so you end up with two cells, 46 chromosomes each, exact same DNA. Okay, so there's just a larger version. Meiosis is a bit more complicated. Meiosis is the process through which you make sperm or eggs, and it's more efficient in some ways. Because in the case of sperm, you're making 4. In the case of eggs, you make 1 egg and then 3 polar bodies, which, to my knowledge, don't really do anything. But, I'm not like a microbiologist, so it could be that there's some function that they have that I don't know. But essentially, you have only one egg at the end. So meiosis only occurs in sex cells. These are specific cells in your gonads. Gonads is actually a unisex term. Gonads includes testicles and ovaries. Okay, so this is occurring in the testicles and the ovaries, and you're making gametes. Okay, sex cells make gametes. Gametes means sperm or egg, and I don't know if you need a mnemonic. Ooh, you got game. It has to do with sexy time. It has to do with reproduction. I don't know. Whatever works for you in remembering this. Okay, so a gamete is either sperm or eggs. Okay, it starts out kind of similar. You've got your chromatin that's hanging out here in the nucleus of the cell. The DNA replicates itself, right? So you now have these X-shaped chromosomes because each of the chromosomes have replicated their DNA. They have twice the amount of DNA that they originally had. Then you get something called crossover or recombination. This is super important because this is one of the absolute major differences between mitosis and meiosis. This is hardcore important, okay? And I will touch on that in the next slide. All right, so you have crossing over, and then you have the first cell division, okay? So that divides into two new daughter cells here. And then you have a second cell division that happens here where this one divides and this one divides. And then you end up with 4 daughter cells, okay? And that goes on to make either sperm or 1 egg and 3 polar bodies. Now, let's just do a little bit of math. When 2 people love each other very much, I'm going to skip a lot of details. You've got sperm and you've got egg meeting, you've got a new baby. That baby, like all humans, at 99, some odd percent, have 23 pairs of chromosomes. Okay, 46 chromosomes in total. So that must mean that the sperm carry 23 chromosomes and the egg carries 23 chromosomes, and then together they make 23 pairs of chromosomes, or 46 chromosomes, right? So that means each of these daughter cells, like each of these sperm cells and the egg cell, they each need to have 23 chromosomes. 23 chrome. And then when they join, then you've got 46, 23 pairs, right? So that's why you have to have 2 cell divisions, okay, is to get the correct number of chromosomes, the correct number of DNA. So you want the amount of DNA that's present here and the amount of DNA that's present here to end up being, like you want this here to be half, right? 23, that's got to be half of the amount of DNA that's here. But then you've got this replication. So here, now, you don't just have twice as much, you have 4 times as much as you need in each one of these sperm cells, you see? So that's why here you have 1 cell division that carries away homologous chromosomes, right? Well, they're not technically homologous, really, because there's been recombination, but I'll get to that later. And then here, each of these chromosomes still is an X-shaped chromosome, right? Which means you have twice the amount of DNA that you want in an individual sperm or egg. So that's why you need a second cell division here, because it pulls those sister chromatids apart. Okay. All right. I will post a video that shows it a little bit more visually. So that'll be on Canvas. So what happens during recombination? Well, if you have any siblings and you're not a twin, you will probably know that you and your siblings are different, right? You may look drastically different or maybe not. You may have quite different personalities. You might be different heights, different eye color, different hair color, different facial features, but you have the same parents. How is that possible? Well, no one really that doesn't boggle the mind so much. You just kind of take it for granted. But there's a reason behind that. And that reason is recombination. So let's say here on the left, you have chromosome 1 from mom. Now, you'll notice this is in the X shape because the DNA has replicated, okay? And here you have chromosome 1 from dad. So these are homologous chromosomes, okay? Homologous chromosomes, meaning, they're both chromosome 1. They both carry instructions for the same areas of the house, although dad may have a slightly different design than mom. Now, recombination, when these two come together, so let's say you are a female and you are making an egg. And so one of your sex cells is going through meiosis. During that process, the DNA that you got from your mom for chromosome 1 joins up with the DNA that you got from your dad in chromosome 1. And your body kind of has to select what you're going to pass on, because you can't pass on both. You're not making a carbon copy of yourself, because your partner is also contributing, right? So, for example, you as an individual, you half of your DNA comes from your mom and half of your DNA comes from your dad. And if you have a child, half of its DNA is going to come from you and half of its DNA is going to come from your partner, right? So you kind of have to, your body kind of has to choose, like, what am I gonna pass on? I can only pass on half. So that's what recombination does. So what happens is these two chromosomes kind of get together and they do their own little, mating ritual. They line up, with homologous portions, and then they swap portions. Okay, so that now there would be another one here also that would look different, like maybe the mirror version of this one. But just to demonstrate what happens is it swaps pieces. So here you get a piece from your mom, here's a piece from your dad, here's a piece from your dad, piece from your mom, piece from your dad, piece from your mom, right? So it becomes, it's like shuffling a deck of cards. And each time it shuffles, you're going to get a different combination because it's random. All right. Now, let's say this is chromosome 1 from your egg, and this is chromosome 1 from your partner's sperm. Later on, your kids are going to have kids. You're going to have grandkids. Okay, so just to track and trace the pieces from the grandparents, right? This is your maternal grandpa, and this is your maternal grandma, right? And here is let's say this is the paternal grandpa and the paternal grandma, right? Well, those shuffle up as well. So then you get something like this in the next generation. Okay, so this is why you're not identical to your siblings unless you're twins. Because each time the shuffle happens, it's random. All right, so there's a larger version of meiosis and mitosis. Now, I tell you what, I'm going to cut this lecture in half because I feel like I've already talked for a long time. So I'm going to come back in the next video and finish this up. So get up, stretch, have a break, refill your water, and I'll be back shortly. See you soon.